pool. And any of the other panelists who wish to put their video on, please feel free to do so. From our organizing committee, the meetings committee, all the people who are trying to make this all seamless in the background and are doing an incredible job. I really want to thank them for all this hard work. So we've been accumulating other questions that have not yet been answered, and we also have 4 pre-prepared questions for the moderated panel discussion. So we'll just try to get through what we can between now and then we have a hard stop at 2 o'clock Eastern Standard Time, so about 28 minutes from now. So let's see what we can do. And again, I appreciate that in the audience, you know, we have people from Europe who are, it's getting kind of late there. So I really appreciate you guys all hanging out with us, including Charlotte. So here we go. So in terms of the moderated panel discussion, the first question that we have for the group is, can we talk about some overarching themes, you know, some take-home conclusions that we can come home with regarding today's session in terms of B-cell responses to SARS-CoV-2. So maybe we could just open this up to various panelists. I could start it off if you like. I mean, I think one of the themes that has gone across multiple presentations now is time. Within the immune response where you are studying the cells that you're looking at, the repertoires that you're looking at, and also the antibodies. And that the time seems to make, you know, not only severity of disease and disease heterogeneity and all the rest of it, but just the time course of infection and how far out you are from exposure to the virus can make a really big difference. And that maybe early in disease course, particularly in more severe disease, you may have more of an extrafollicular response that's dominated by large clones and plasmablasts, and perhaps relatively low levels of affinity maturation, and in severe patients, perhaps an impaired BT collaboration. But that if you you know, get over the infection and then your subsequent immune response may be very different, right? So that's one, I think, overarching theme. Does anybody want to kind of extend that and talk about that some more, or pull out another overarching theme that you'd like to comment upon?
Actually, so I was pretty happy to see that one overarching theme between both your and my talks, you know, was the contribution of plasma cells seemingly to the response and the increased frequency of those, especially looking at this early wave of unmutated plasma cells and large clonal expansions. That seems to be a pretty consistent feature of what we've seen in the repertoires. As well as, I didn't present this, but we also found evidence of increased CDR3 length in COVID patients. So that's definitely one thing.
Yeah, and you know, Brian, maybe you could comment on this in terms of other viral infections, since you have a long history of studying viruses, probably longer than most of the rest of us. In terms of, you know, is this really kind of just what the immune system does in the beginning, right? It's just mount this massive, you know, plasmablast, all hands on deck style response, take whatever's there and go with it.
Yeah, it seems yes. So it's a little difficult to study for a lot of reasons, but so in Ebola, for example, Scott Boyd had a lot of really good data about the initial response to a primary infection. So you have a lot of class switched but unmutated plasmablasts that tend to be weakly functional, if they're functional at all. When we did some work on Zika a couple of years ago, the plasmablast response was quite good. We saw a lot of neutralizing antibodies in that plasmablast response, and we saw some clonal expansion, although the most clonal expansion and the best and most mutated antibodies were in people that had previous dengue infection. So again, you're getting cross-reactive recall more than you're getting kind of a rapidly mutating primary response. But the problem is a lot of the data that's out there about plasmablasts tends to be in flu vaccinees, and you're not at all really getting a primary response in those patients. So a lot of people are saying, well, yeah, plasmablasts, you get great antibodies from plasmablasts. And that's true if this is a subsequent exposure. It doesn't seem to be as true if it's a primary exposure.
Yeah, so that's a really key point, is that this virus, of course, is different from flu because we haven't seen it before, other than SARS-CoV-1, which is a little bit of a one-hit wonder, right? So a very small proportion of the population has seen that. And then, of course, MERS, which, again, very small proportion of the population. The other beta coronaviruses have some degree of similarity, of course. And so those are the people that we're most interested in finding, right? And so your one donor who you really wanted to get antibodies from, who probably had a nice, titers of antibodies to OC43 or one of the other beta coronaviruses, would have been very, very interesting. Yeah, and I think in terms of that's another potentially a theme that goes across different talks, right? Is the notion of cross-reactivity and multi-reactivity. And as we learn more about antigen-specific cells, what does that really mean, right? So will we be able to define the characteristics of antibodies that can see, that might be the sort of holy grail of virus research, right? The so-called broadly neutralizing antibodies. So do people want to comment on that at all in terms of what properties such antibodies might have, and whether that can help us, help inform vaccines or also, for that matter, passive immunotherapy? No takers on this one? It's a tough question. And there's a lot of questions wrapped into that one, right? So maybe we could think a little bit more about sort of what are the properties of an ideal antibody that you might want to have as a therapeutic.
I think SARS-CoV-2 is a little bit unique in the sense of broadly neutralizing, because there aren't a lot of people, at least that we've looked at, that have SARS-1 exposure followed by SARS-2 exposure, or MERS followed by SARS. So there doesn't seem to be a lot of pressure on the immune system to make broadly neutralizing antibodies. And SARS-2 doesn't seem to mutate very rapidly anyway. So there doesn't seem to be a lot of pressure put on the immune system to focus on these highly conserved epitopes. I think if we wanted to identify these, I think it's possible, but what I think would be the easiest way to do it is find the epitopes that you think are most likely to be conserved across these viruses, and then try to pull out antibodies against those. But again, if they're not immunogenic, you may not find many of them around. So again, since there's not much pressure to focus on those epitopes, I think you're being driven by immunogenicity more than by, viral variability, I guess.
Yeah, no, that's a key point. Charlotte, I see you nodding your head. Did you want to add to that or comment on that in terms of your database and how we might?
I was going to say almost exactly the same things that Brian's just said.
Yeah.
The other part of your question that you were talking about there, about in terms of what we'd want from a therapeutic, I think there are lots of properties now that you're able to test for just from knowing the sequences of these antibodies about whether they'll make a good therapeutic. So there are obvious things like if we can have a shorter H3, that tends to make life easier. But we've done, and so have others, quite a lot of work to try and think about that very early on in terms of what makes a good therapeutic, because it isn't as simple as it's a nice human antibody. It also depends how it would behave if we produced it in large concentrations and all those kinds of things. So a lot of what's currently being pulled out as potentially useful might not translate into like a passive therapy you could use in any way, because you just couldn't keep it in a bottle on a shelf well. And I think that's something we need to start doing very quickly as we start thinking about these pieces as well, to be able to move that way.
Yeah, Brian, did you want to comment on that in terms of your ongoing efforts? Do you have a sense of whether there are certain sort of lead candidates that are emerging, not necessarily your efforts, but actually just globally perhaps also, for the kind of antibodies that are ideal in this respect?
Yeah, I think a lot of what Charlotte said is exactly right. You want primarily you're going to want antibodies that are tolerable and elicitable, right? Depending on whether you're looking at a therapeutic or you're looking at a vaccine, right? So if you're focusing vaccine development, again, you'd want shorter CDR3s, because that's where the immune system tends to live, is in that like 12 to 16 kind of range is where most of the action is. So if you're trying to elicit something like that, you're probably going to have more success than if you want to elicit something that has 28, 29 amino acids long, right? Those are just rare to start with. So it's going to be harder, just stochastically, it's harder to find those things and get them going. And again, for a therapeutic, there's obviously a lot of other concerns, manufacturability, all of these things too that you can pile on top of it. But yeah, I think at this point, I don't know how much of a benefit breadth is going to give you, because there doesn't seem to be a lot of kind of global diversity in SARS-CoV-2. There's some, but not a lot, nothing like flu or nothing like HIV. So it doesn't appear that we're getting a huge amount of escape from potential therapeutics. So until we see that, I'm less worried about it in terms of breadth stuff. Yeah.
But in that, along those same lines, what do you see as the role for passive immunotherapy in SARS-CoV-2? How would that work exactly? I mean, yeah.
I'm like a huge antibody guy in general. So I like if anyone was going to be like, like leading the band for antibody immunotherapy, it would be me. I think I don't want to say there was a window, but I think the early success of vaccines that have been moving so quickly is kind of closing the window. I mean, I would see the window of really widespread use of antibody therapy as the gap between when we can find antibodies and the point at which we have a vaccine that's safe and works well, right? So I don't know why you would be giving people, pre-exposure prophylaxis, for example, if there's a vaccine, right? That doesn't seem like that'd be, unless you're in a high-risk population that doesn't respond well to vaccines, but now the market is very small once a vaccine's available. So my gut feeling was that antibody as a therapy would be that bridge that gets you from we don't have any drugs to like we have a vaccine or we have a really good small molecule drug or something that's less expensive than an antibody. So even though I love antibodies, I would hope that they aren't even needed because the vaccine's so fast we don't need to worry about it, right?
Right, but you raise a really good point, which is that, I mean, the vaccine can certainly help with reducing spread, and if you can have herd immunity that you at least induce in the part of the population that can respond robustly to the vaccine, that's great. But the people who are at greatest risk for the disease are actually the elderly and people with these pre-existing conditions that can compromise their immune system, as well as other concerns. And in those populations, you might not see a very robust immune response. And so there, having some other passive immunotherapy might make sense, particularly if those populations are in some ways isolated or cloistered together. Like, you can imagine in an old age home or something like that, where, even though you don't have spread from maybe the younger generation to the older generation, you might have spread from the older generation to itself, right? So that's a really interesting thing to consider, I think.
Can I just add in on there on the antibody therapy? Because I think I agree with what's been said, but I think it will end up being an important part of the overall response here, in terms of, I think a lot of people still talking about the idea that we will be eradicating COVID, and I don't think that's a correct way of thinking about it. Given the level of pandemic spread we've now seen, I think we will be living with it, and an effective vaccine will mean that, in many areas we can deal with that, but it will take a long time before we've managed to vaccinate enough of the population. And the other problem probably with the vaccines is, at the moment, the indications would suggest that they're not going to be something I have once and I never need the vaccine again. And so I suspect that antibody therapy and small molecule therapies are going to be a big part of a kind of global picture. I think it's a much smaller, I agree, I don't think they're going to be what we need for everybody, but I don't want people to think we shouldn't be developing them, because actually I think we're going to need them as one of the tools in our armory going forwards. So I don't think they're going to be as key as they might have been had vaccines looked like they were much harder to develop, but I still think they're going to be a key tool that we're going to need in order to basically live with this as a disease that will be racing around the population, I suspect, for a very, very long time now. That wasn't that cheerful, was it? Sorry.
Wow, what a depressing comment. But yeah, I think, go ahead, Brian. Brian, you're about to say something.
I just said exactly what I was trying to say, much more succinctly and clearly, is that I think there was a time when everyone was thinking every first responder is going to get prophylaxis as antibodies and all of this. And I think that may be happening less and less. But yes, I agree. Antibodies are definitely an important part of it, but I don't think it's going to be, there was a time when I think we all thought that was going to be what was the bulk of the response. Like that's our hope of slowing the spread and all of this stuff. I think it may be less of that as antibody therapy, but yes, I think Charlotte's exactly right.
Yeah, so genie's out of the bottle, as it were. But on the other hand, we don't yet know what the longer term picture will look like, right? And so how well will we be protected from future infection if we were infected the first time, let alone receiving a vaccine, right? So that's it's tricky right now. There's so much we don't know. And in that connection, if we're thinking about sort of monitoring people for infection and trying to figure out how to get the population back to work, do members of the panel have any thoughts about how we should try to be doing that in terms of any of the B-cell based assays? Any thoughts?
I think at the moment we're not dead certain that any of the B-cell based assays would give us a clear indication that, you know, somebody was protected or safe in any way. So I think it's a very difficult question. I also think there's an awful lot of, even if we did know that, there's a lot of social science around this about behavior change and what happens when you make those kind of decisions that makes me a bit nervous. However, having said all of that, I think the development of these B-cell based kind of testing would be a really good way forwards in terms of many things we would hope to be able to analyze and look at within the population. But whether they can be used successfully in terms of helping people live their lives might be a bit more difficult. I keep being told by social scientists we have to be very careful about this. So in the other half of my life where I work with government on this problem.
Yeah, no, that's really, I think you're very right. And we have been very conservative in what we've suggested that people do, right? So, so for example, you know, in Pennsylvania, a lot of the assays that are being used on individuals who may be infected are, you know, are PCR-based assays that look for viral RNA. And that makes sense, right? That you would maybe look for RNA or for an antigen-based assay to determine whether somebody has virus currently, because that's really the thing you're trying to prevent. You're trying to prevent spread of somebody who's infectious. And then you're right that the correlates of immune protection so far for serologic testing are not yet well defined. Although we're starting to get some inkling that we should be able to maybe use some of these assays in this way. And as we get more long-term data, maybe that will in fact happen. So one of the things that I think is interesting to consider is using serology as kind of a get out of jail card eventually, when you have a population that's very closely monitored. And if you have somebody who previously had infection and now has antibodies and doesn't have detectable virus, you know, what do you do with that, with somebody like that? And the concern from the social sciences side is if you release somebody like that back into the general population too early and they relax on all their social distancing, then do you create sort of, you know, super spreader events from doing that? So it's tricky, but maybe with combined assays, right, where you use a viral antigen test, or if we can get those to be sensitive enough, plus an RNA-based assay, and then serology for sort of longer term follow-up in individuals who did previously test positive. So the follow-up there is interesting, because some of what we learn about the breadth of the immune response as well as the isotype usage may give us an idea about timing. So that's one of the things that I thought was so interesting about that study that came out of the Sorbonne, was that IgA antibodies to the virus seem to be shorter lived than IgG. And so could one consider sort of a strategy where you do a high sensitivity screen for an abundant antigen like nucleic capsid, for example, that seems to be highly immunodominant and easy to detect by serology. And then you take people who are positive in that screen and you run them through an RBD assay. And maybe you run it with RBD for IgG and IgA. And if you see that they test strongly positive for IgG and IgA, you say, this person's probably still pretty early in their disease course. Whereas if they test positive only for IgG and not IgA, maybe they're a little bit further along. So those types of assays for other viruses are well known, right? That by serology, you can kind of time certain aspects of the disease. Of course, for SARS-CoV-2, we're still trying to learn all of that. And then the added dimension of difficulty is then with the vaccine, is figuring out what kind of assays to use to monitor adequacy of the immune response to a vaccine. So in that connection, do any of you have any suggestions for what one might do in terms of either the antibodies, the isotypes, or for that matter, the immune repertoire features. Do you envision any type of assay for that?
At least in terms of the immune repertoire, it seems like, at least coming from some of the talks that we've seen today, that a lot of the features of BCR repertoires from SARS-CoV-2 infection are pretty similar to what we might expect from just regular viral infections or other viruses. So I think it might be difficult to define some sort of repertoire feature that's specific. Maybe convergent antibodies or testing for those.
Also, I think one thing that could be nice that comes out of a lot of it, since we're doing these huge vaccine trials, is maybe some level of serum antibody that does correlate with infection or protection better than just, 2x what convalescent patients tend to have, right? So we don't even know if that's protective. Maybe 10% of that is actually protective, right? You could be pretty low level. And also, I mean, we don't really care about serum antibody or plasma antibody. We care about antibody at the mucosal surface, right? So in theory, circulating antibody should correlate pretty well with mucosal antibody, but and also I don't think we necessarily need sterilizing immunity against COVID-2, right? If we just have enough to slow it down that you don't get symptoms, I guess there's technically the potential for spread if you're an asymptomatic person, but I think for most cases, sub-sterilizing immunity that makes every infection asymptomatic would be almost as good as complete protection, right? So yeah, there's a ton to figure out that we just don't know. So giving someone an antibody test and saying you're good, you can go, go out and do whatever you need, I think it's going to be a tough sell. Hard, I think, yeah.
Yeah. No, I hear you. I wanted to mention, I've been monitoring the Q&A here, and there's a comment here from Thomas, excuse me, saying we find heavily mutated IgA expanded clones among plasmablasts. However, none binds to SARS-CoV-2 proteins upon recombinant expression. So that's a really interesting observation. So Brian or Charlotte or Ken, have any of you seen similar things in your data?
So a lot of our monoclonal stuff was IgG only, just because those are much easier to express, and you're not then expressed recombinantly, and you're not, relying on some feature of IgA, dimeric IgA. You may not get the same function if you just put that into an IgG expression vector, that kind of stuff. So we focused on IgG. But again, I think it also comes down to the quality of our SARS-2 proteins, right? Do they mimic native stuff? It's hard to make an IgA recombinantly and test it for neutralization, but that would be, I think, the ultimate readout. If you just look at the protein itself, there does seem to be a lot more conservation between SARS-1 and seasonal coronaviruses and MERS, kind of towards the base of the spike. So if the IgAs tend to recognize that region, my gut feeling is that that's the least ordered region in our soluble constructs. So you may see the most disruption there. Yeah,
that's interesting.
A ton of highly mutated IgA. We see them from our 10x data. They're mutated about as much as you see IgG mutation. So it's not a special. I mean, it's a little higher, but it's not like it's twice as much.
Right, and I think in the early stage of infection, it might be that a lot of the IgAs that are mutated may be getting recruited from pre-existing memory clones, where if you look at mutations of mucosal antibodies just in healthy individuals, we've looked at that in organ donors, the antibodies that you can find in the GI tract, for example, which are largely IgA, tend to have higher levels of somatic hypermutation sort of at baseline. So that it may just be a selection bias in terms of which clones you're pulling out of your IgA repertoire. But that's super interesting, and I think that it does bring back the idea of which antigen, you get out what you put in. And so so have you looked, Brian, at any more complex antigen systems? Like, have you tried looking at whole virus or VLPs or anything like that in terms of trying to isolate antibodies, or do you think that that's just going to make the problem 100 times worse, because now, in addition to all the stuff that's sticking to the spike that's wrong, now you've got multimerization going on?
Yeah, so for our single cell sorting stuff, I mean, speed was like the priority, right? So we wanted to get a paper out fast. And it tends to take quite a bit more optimization in terms of flow to use something like a whole virus, because they tend to be sticky, and there's a lot of other things. So you end up sorting a lot of things that end up not being the function you want to get the things that are the function you want. My general feeling is that if you want to do like whole virus type stuff, it's more easy to do, or it's easier to do something like a B cell stimulation and then test the secreted antibody. So kind of like the Mark Connors method, or what Theraclone did, like the single cell stimulation, or what like Lanzavecchia's immortalization stuff. I think that's the easier way to test things like that, because you're not testing like a big multimeric thing against another big multimeric thing. You're testing a recombinant antibody or a single antibody against a multivalent thing, which can be cleaner. But that's we haven't done much of that yet. That's outside of our primary focus.
Fair enough. So we have about 2 minutes left here. I just wanted to maybe go around to the speakers and let each one say any last remarks for the day. So, so Ken, if it's all right with you, why don't we start with you? And is there anything else you'd like to say to the group?
I didn't prepare any last remarks.
No, we don't. This is all free form, right? So you don't have to say anything if you don't want to.
Yeah, I guess just to answer the question about IgA. So we do track that in our studies, but if we look at our expanded clones, I'm just looking at the figure now, they're composed mostly of IgG and IgM with a little bit of IgA. And that might be because we sampled from the blood. So that's,
fair enough.
Yeah, but I would say that in our data, at least, the expanded clones don't seem to be IgA.
All right. Okay. So Charlotte or Brian, do either of you have any last words for us?
I will go really quickly, which is I want to say thank you for inviting me, and it was really good fun to listen to everyone else's talks as well. And the other thing is just my generalized plea: if you have seen papers that are relevant for our COVABDAB database, please tell us. And that continuous plea about making this data available and keeping it out there, because I do think like the vaccine is going to be incredibly important. Understanding the response to that, understanding how people respond in terms of these repertoires, is one of the key things we need to do. And as a community, we shouldn't forget how important that might be. And in the same with the antibody therapy and all the other things. So please, everyone keep going, because I don't actually carry out the experiments. So it's really important to me that you guys do, because then I can do something useful, hopefully, with the data. So thank you, and please keep doing it.
Well, I think that's if that's okay, Brian, well, we'll just cut it off here. We're at 2 o'clock, and I want to also echo Charlotte's comments. This is a communal effort, and that's what the AIRR community and all of us on this call today are all in favor of this. So we work together. I think COVID has shown that we work together in a really spectacular way. That science, that this, of all the awful things this virus has brought us, the one sort of shining good thing it has brought us is this, is that the scientific community has really come together and work together in very creative ways. And so I hope we can all keep that up. And so we're going to convene again tomorrow and talk more about how we can work together. So again, thank you so much to all the panelists, and be well, everyone, and we'll see you again tomorrow. Take care.
Thank you.